Okay, welcome to the fourth lecture in Bio 1012 lecture series, and today I will be covering protein synthesis. As usual, let's go through the course objectives and lesson objectives. This lesson comes under uh, functioning of different cell structures, and at the end of this lecture, you should be able to describe the process of protein synthesis and explain the function of different enzymes in protein synthesis, and when you are given a code, you should be able to ascertain or determine the amino acid sequences. In central dogma of molecular biology explains how information flow from DNA to protein. So this concept was put forward by Francis Crick, the one of the scientists who contributed to the identification of the structure of DNA. So in biological systems, information is stored in DNA, then that information is passed down to protein and expressed as phenotypes. So in central dogma, the information in DNA is passed down to RNA. This process is called transcription, and the information in RNA is passed down to the protein, and this step is called translation. And you can see there are two arrows going, circular arrows, which means DNA can pass down its information to other DNA molecules. This is what happens when DNA replicates. RNA can do the same. RNA can produce new RNA molecules. So information in RNA molecules can be passed down to the resulting RNA molecules. And back in 1970s, scientists found that RNA can contribute to the formation of DNA. So the information can move from RNA to DNA as well. Okay, right. Now let's briefly look at the structure of a DNA molecule. So in a DNA molecule, we have a sugar group. We call it deoxyribose sugars, and in RNA molecules, instead of deoxyribose sugars, we have ribose sugars. So in DNA, the difference is there is a one oxygen group missing in the sugar molecule. Then we have a phosphate group attached to the sugar molecule, and we have two types of bases, purine and pyrimidine. In DNA, the two pyrimidines are cytosines and thymines. They are six-membered ring compounds. And in RNA, instead of thymine, we have uracil. Then there are two types of purine bases in DNA molecules. Those are adenine and guanine. So these are much larger than the pyrimidine bases because other than the six-membered structure, there is a five-membered structure attached to the six-membered structure. DNA molecules form double helix, but in RNA they only form a single strand. So these in DNA, the double helix is bind together by hydrogen bonds between the nucleotide bases, as you can see over here. Right. So when we talk about the flow of genetic information, the information is stored in the DNA in the form of specific nucleotide sequences. Okay. the DNA inherited by an organism inherit that information from their parents, and that particular information dictate the synthesis of proteins. What type of proteins are produced is determined by that particular information. So the resulting protein act as the link between the genotype, or the information stored in DNA and RNA, and the phenotype. So this expression or phenotypic expression can be explained in the process called gene expression. So gene expression is the process by which DNA directs the synthesis of proteins, and in certain cases RNA can contribute to this process as well without the involvement of DNA. So I'll take a very famous example. I hope you know Mendel's pea experiment. So in his experiment, he observed two types of pea seeds: round seeds and wrinkle seeds. So when the genotype is homozygous dominant or heterozygous, the plants produce round seeds. If the plant produce if the plant is homozygous recessive, the plant produce wrinkled seeds. Right. So when we talk about DNA stores genetic information, it stores this information in certain part of the DNA molecule. We call them genes. If you take a piece of DNA, they are double stranded, and you can identify different genes. Here in this image we have 3 genes. So the code for protein synthesis is embedded in this region. So in a DNA we have 2 strands, we call them antiparallel strands, bind together by nucleotide, complementary nucleotides, and only one DNA strand is used in the synthesis of protein for a given gene. If you take gene 1, in this case only this top strand is involved in the synthesis of the protein. That doesn't mean only the top strand in a given DNA molecule contribute to protein synthesis. In a different location, for example, we'll take this gene number 3. In gene number 3, the bottom strand may be involved in the synthesis of proteins. But for a given gene, only one strand is used in protein synthesis, and we call it as the template strand. So the information in the template strand is transcribed to an mRNA molecule, and the information in this mRNA molecule is translated to the protein. So this is the basic process how information in DNA is passed down to the protein level. So let's look at the structure of a gene. So a gene is a region of specific nucleotide sequences in a chromosome that codes for a specific polypeptide chain. And as you can see here, the coding sequences, we call them exons, are interrupted by non-coding sequences called introns. And in a gene you will find other regions as well, enhancer regions, both in upstream and downstream, a promoter region, and a very unique area called the Tata box. Okay, so in a gene you have, remember, you have both coding and non-coding regions. So when we talk about protein synthesis, it takes place in 4 stages. The first stage in protein synthesis is known as transcription. Second stage is the RNA processing. The third stage is translation, and finally the fourth stage is post-translational processing. So I will discuss these stages in detail. Okay, so we'll start with transcription. So there are 4 stages in protein synthesis. If you take the first stage, which is transcription, there are 3 stages in transcription as well. So what are these 3 stages? First step is initiation of transcription, elongation of the RNA strand, and the termination of the transcription. So we'll start again with the first step of the transcription, initiation of transcription. The promoter signals the initiation of RNA synthesis. So information in the DNA should be passed down to RNA molecules during transcription. So the promoter signals this initiation. There are factors called transcription factors, and these transcription factors mediate the binding of RNA polymerase, an enzyme involved in transcription, and it initiates the transcription stage of protein synthesis. The assembly of transcription factors and RNA polymerase II. RNA polymerase II is the enzyme involved in this process, bound to a promoter called transcription complex. So formation of the binding of polymerase II to the promoter called transcription complex is important. And there's another promoter called Tata box, as I mentioned in the previous slide, is very important or crucial for the formation of initiation complex in eukaryotes. So what are these transcription factors? So transcription factors are the proteins that binds to the DNA near the start of transcription of a gene, and they can either inhibit or assist RNA polymerase in initiation and maintenance of transcription. So the activity of RNA polymerase is controlled by these transcription factors. Okay, so here we have the promoter region with the Tata box, and here so these are the 2 DNA strands, 5 prime and 3 prime, and in eukaryotes we have a promoter region called the Tata box. So transcription factors first binds to this Tata box prior to the binding of RNA polymerase II. Okay, then once those transcription factors are bind to the Tata box, RNA polymerase II can bind to the DNA molecule forming transcription initiation complex. So this large structure here is the RNA polymerase 2. So once the RNA polymerase 2 is bound to the DNA, forming the transcription complex, RNA synthesis can take place. You can see here RNA is now transcribed. Okay, let's go through this once again. Here's the region of the gene where we want to transcribe. Now we have the transcription initiation complex, including the RNA polymerase. So once the RNA polymerase 2 is attached, it starts to unwind the double-stranded DNA, and one strand of the DNA is used as the template for the RNA molecule. So the strand 3' to 5' is used in this transcription stage, okay, not the 5' to 3' DNA strand. So now the first step, the initiation of transcription is done. Now the second stage in transcription is the elongation of the transcription. So as RNA polymerase moves along the DNA strand, it opens up or untwists the double helix, usually 10 to 12 base pair at a time, and transcription or formation of mRNA molecule take place at a rate of 40 nucleotides per second. So for mRNA molecules to form, you need nucleotides in the cytoplasm. By the way, actually this stage takes place in the nucleus. Okay. If you take a certain gene, several RNA polymerases can transcribe that gene at the same time. So for example, one RNA polymerase can transcribe a particular gene followed by another one, because we need more copies of mRNA in order to produce the protein. I mean, by producing only one strand of mRNA is not enough. Higher the quantity, faster the formation of protein. Right. So as the RNA polymerase 2 moves along the DNA strand, RNA strand is produced. We call this as the mRNA molecule. So the resulting mRNA molecule is 5' to 3'. So if we take a closer look at of this transcription area, you can see we have two DNA strands near the RNA polymerase is attached. The two strands are unwound or detached, and the 3' to 5' strand is used as the template DNA strand, and new RNA nucleotides are attached to the complementary RNA nucleotides are attached to the resulting RNA molecule. So as the RNA polymerase moves along the DNA strand, more and more complementary RNA nucleotides are attached, and you will get a mRNA strand. So at one point this transcription should be stopped, and we call that step as the termination of transcription, termination in end. So in eukaryotes, polymerase continues its transcription after the pre-mRNA. The resulting mRNA is cleaved from the growing RNA chain, and eventually the polymerase will fall off from the DNA. So, I mean, imagine you have a gene about this long. So the RNA polymerase moves along the DNA molecule, keeps producing the transcribing the mRNA molecule, and it keeps going on, and at one point it falls off. So you can see here. So once the end is reached, end of the gene is reached, after some time the RNA polymerase is detached. So now we have a mRNA molecule called pre-mRNA. Okay. Before we use this pre-mRNA molecule in the next stage, which is translation, this pre-mRNA molecule should undergo certain modifications. Because if you remember the structure of a gene, there are exons and introns. I mean, we don't need the introns. We only need the information in the exons, right? So the enzymes in the eukaryotic nucleus modify these pre-mRNA molecules before they are dispatched to the cytoplasm where translation takes place. So remember, transcription takes place in the nucleus, and the mRNA molecule should go to the cytoplasm to complete the translation process. So prior to that, RNA processing should take place. So during RNA processing, both ends of the pre-mRNA is altered. So if you take a mRNA molecule, pre-mRNA molecule, 5' you have 5' and 3' both ends are altered. And as I mentioned earlier, some of the non-coding parts are also cut out and spliced together, so that we only have a piece of mRNA containing whatever the useful message to the formation of the protein. During this RNA processing, 5' end receive a modified nucleotide cap. We call it a 5' cap. The 3' end gets a poly A tail, polyadenylene tail. We call it polyadenylation. So why do we need these changes, this capping and the splicing, especially these two, this modification at the to the ends? So it seems like these modification facilitate the export of mRNA molecules from nucleus to the cytoplasm where translation is taking place. And these modification protect the mRNA from hydrolytic enzymes, because the enzymes in the cells can degrade or digest these resulting mRNA. If that happens, protein synthesis cannot take place. So having these modification prevents these mRNA molecules being broken down. The 5 prime cap helps the ribosome to attach the mRNA molecule. So, ribosomes are involved in the translation process. So, this 5 prime end helps that attachment. Right. So, here we have a modified or processed mRNA molecule. You can see at the 5 prime end we have a 5 prime cap containing phosphate groups and guanine nucleotide. Then there's a region, call it so UTR, untranslated region, and we have our start codon. So the start codon is important during translation, and we have our protein coding segment. This is where the information is stored in, and we have a stop codon. So during translation stop at this point. Then we have polyadenylation signal. Again, this region is again not translated, just like this 5 prime UTR region. And at the 3 prime end we have poly A tail. So poly A tail contains 50 to 250 adenine nucleotides attached to the 3 prime end. So these are the two modifications, poly A tail and 5 prime cap. So poly A tail is added to the 5 prime as the RNA polymerase transcribe the DNA, and here I have the structure of a 5 prime cap. I don't think you will see this because of my inlay, but in the lecture note please refer this figure. Okay, and here we have the phosphate group. Okay, right. So in eukaryotes we have non-coding regions, between coding regions. These non-coding regions are called introns, and we have to get rid of them before we use these mRNA molecules in translation. Okay. So this removal of introns and joining of exons is called RNA splicing. So once splicing is done, we have a mRNA molecule with continuous coding sequence. So here we have our modified mRNA, pre-mRNA molecule containing introns, non-coding regions. So once splicing is done, the cell get rid of these introns and join these regions together, forming a continuous exon or information coding mRNA molecule. So once splicing is done, we have our 5 prime cap, coding segment, and the poly A tail. So let's see how this is done. So this splicing is done by structures called spliceosomes. So Spliceosomes are made out of proteins and several small nuclear ribonucleoproteins or SNRPs. These things, small RNAs. So, if you remember in the previous lecture, I told you that RNA can act as enzymes because they can uh, uh, couple, pair up, the bases can pair up uh, by themselves by finding complementary segments, and eventually you'll have three-dimensional RNA molecules with something called sticky ends, right? We'll get to one such structure in a bit, the tRNAs, and so these RNAs and proteins form these spliceosomes. What they do is they attach to these beginning and ends of introns, and they cut the introns at these exon regions and stick these um, exons together. For example, I don't know whether you can see this properly. Imagine you have a piece of intron. What this spliceosome does is it forms a loop like this and cut from these two ends and join the two exons, forming a continuous mRNA molecule. So let's see how it's been done. Here we have exon 1 and exon 2 separated by an intron. We have uh, sRNA molecules and other proteins. They bind together, forming a spliceosome, and remove the intron region and join the two exon regions or protein coding region. Right. So now we have a modified mRNA molecule, messenger RNA molecule. So synthesis of messenger RNA molecule take place in the nucleus through a process called transcription. Then it undergoes mRNA processing. Then these processed mRNA molecules enters into the cytoplasm for translation stage. So translation is the second, actually the third stage of protein synthesis, where ribosomes read the genetic message in these mRNA molecules and use that message to code for proteins or polypeptide chain. Okay, now you can clearly see how the information flows in this system. Information which was in the DNA was passed down to the mRNA, and through mRNA processing, non-coding regions were removed, and that message is now in the mRNA, and that information in mRNA is translated to proteins where they perform various functions. Okay. So let's see what are ribosomes, because ribosomes are the structures involved in translation stage. So ribosome facilitate specific coupling of tRNA. What are tRNA? tRNA are the transfer RNAs. They facilitate the coupling of tRNA anticodons with mRNA codons in protein synthesis. Ribosome has two parts, large subunit and a small subunit, and there are three sites, we call them E, P and A, and This is the structure, and here we have our mRNA molecule. This is the structure which used in the translation process. The two ribosome subunits, the large and small subunits, are made out of proteins and ribosomal RNA, or we call them rRNAs. Okay, so this is the structure involved in translation. So, a given ribosome has multiple tRNA binding site. We call them E, P, and A site. If we take the P site binds the tRNA attached to the growing peptide chain, or we call this site as peptidyl tRNA binding site. Then we have A site. It binds the tRNA carrying the next amino acid, which is going to be attached to the polypeptide chain, or we call this as amino acyl tRNA binding site. Then finally we have the E site, or the exit site, which binds the tRNA that carried the last amino acid. So once the amino acid is transferred to the which was on the tRNA transferred to the growing polypeptide chain, the tRNA moves to the E site and get detached from the ribosome. The ribosome has two primary functions. It decodes the message in the mRNA, and it forms the peptide bond, which means it reads the message in the mRNA molecule, and according to that message, polypeptide bonds are formed to make the peptide chains, polypeptide chains. The enzyme peptidyl transferase is the enzyme used by the ribosome to form bonds between amino acids. So for translation to take place, we need tRNA molecules or transfer RNA molecules. So these transfer RNA molecules carry amino acids to the ribosome for incorporate into the polypeptide chain. Amino acyl tRNA synthetase add amino acid to the amino acid attachment site of the tRNA molecule. In a given tRNA molecule, I will explain the structure in a bit. There is a region called anticodon loop. So this anticodon loop contains 3 nucleotides complementary to the mRNA codon. Okay, so here we have a structure of a tRNA molecule. Usually tRNA molecules are 80 nucleotides in length, and you can see there are regions where these RNA molecules are coupled. So the complementary regions can form H bonds just like in a DNA molecule, and they form this pairing up. So this pairing of nucleotide bases can give rise to this 3D structure. And in a tRNA molecule, at the 3' end we have amino acid attachment site, and at the 5' end we have a phosphate group, and there are 3 loops. First one over here is called the D loop. On the other side we have a T loop, and we have another loop where it carries the anticodon. So anticodon, as I mentioned earlier, contains the nucleotide bases complementary to the mRNA molecule. So depending on the anticodon determines what type of amino acid is attached to this tRNA molecule. Okay. So here we have a three-dimensional structure. So it's not a plain structure, it's a twisted three-dimensional structure. So when we talk about amino acids, in a given cell we have 20 different types of amino acids. You can group them into 4 categories. There are non-polar amino acids, polar amino acids, charged amino acids, both positively charged amino acids and negatively charged amino acids. We have 20 different amino acids, but we only have 4 nucleotide bases. I mean, if a given nucleotide can attach to one type of amino acids, there can be only 4 types of amino acids in a given cell. If we think 2 nucleotide bases form a given amino acid, then we can only have 4 to the power 2, which means 4 times 2, 16 amino acids. But we have 20 amino acids. But if 3 nucleotides contribute to a given amino acids, we can have 4 to the power 3, which means 64 different types of amino acids. But we only have 20 amino acids, so 3 nucleotide code is more than enough to create these 20 amino acids. We'll see how it's been done. So when the tRNA molecule or the ribosome reads the mRNA molecule, reading of the mRNA molecule is done in codons, in 3 nucleotide base pair segments, right? So in the tRNA molecule, we need 3 nucleotide base pairs which are complementary to this mRNA molecule, right? So we have codon tables. Actually, I mean, if you read a textbook, you will find a table for codons for mRNA. But I have used this image here, this circular diagram. It does the same. You start at the center, so that's the first nucleotide. Then the second ring is the second nucleotide, and the third ring is the third nucleotide. For example, for methionine amino acid, the codon for methionine is A, U, and G. Adenosine, uracil, and guanine. If you take something like tyrosine, you can have 2 different codons, UAU or UAC. For serine, you can have 1, 2, 3, 4 different types of codons. So any of these codons, any of these 4 codons can code for serine amino acid. And there are 3 codons UAA, UAG and UGA which act as the stop codon. So, in our mRNA strand at the very end we have this stop codon. Any of these 3 stop codons, either UAA, UAG or UGA in the mRNA strand at the 3' end, so that the ribosome knows, okay, this is the end of the coding region. Right. So, let us see how translation is done. If transcription is the first stage, transcription means writing. So, you are writing the information in the DNA into the mRNA. Then the second step is the translation or reading of the mRNA strand. So, here we have a ribosome attached to a mRNA molecule. So, this attachment takes place at the 5' end. So, as the ribosome moves along the mRNA molecule, the message in the mRNA molecule is read in codons or 3 nucleotide base pairs, and the tRNA molecules brings the corresponding amino acid, and the amino acid carried by the tRNA molecule is passed down to the growing polypeptide chain, and a polypeptide bond is formed, a peptide bond is formed, forming a long chain of amino acid. Similar to transcription, translation also has 3 stages. First, initiation of translation, elongation of the polypeptide chain, and termination of the translation. So, we will start with the first stage, initiation of translation. So, in the initiation stage, we need the mRNA molecule, tRNA molecule with the first amino acid and the 2 ribosomal subunits. First, the small ribosomal subunit binds with the mRNA molecule and a special initiator tRNA. Then the small subunit moves along the mRNA molecule until it reaches the start codon. The start codon is always AUG. So, as the small ribosomal subunit moves along the mRNA molecules, it looks for this AUG code. Once it found that AUG code, it stops there. Then proteins called initiation factors brings the large large ribosomal subunit and completes the translation initiation complex. Okay, here we have the small ribosomal subunit. It gets binds to the 5' end of the mRNA molecule. Then it moves along the mRNA molecule until it finds the AUG codon. Then we need the tRNA molecule carrying the methionine amino acid, and it binds to the AUG region of the mRNA molecule. Once that attachment takes place, the large ribosomal subunit gets attached and complete the translation initiation complex. So, for this to take place, we need energy. So, energy is used in this translation stage. The next stage of the translation is elongation of translation. So, during elongation, amino acids are added one by one to the growing amino acid or polypeptide chain. And each addition of amino acid involves elongation factors and occur in 3 steps. Codon recognition, translation is done in done by reading 3 base pairs at a time, or we call them codons. That's the first one. First, the codon must be recognized. Then a peptide bond must be formed, and the third step is the translocation. Right. So here we have a growing amino polypeptide chain. You can see here a tRNA molecule bringing in another amino acid. If this amino acid, if this tRNA has the anticodon complementary to the mRNA codon, this tRNA molecule will get attached to the A site, which requires energy, and it recognizes that mRNA codon. Then the tRNA molecule transfer its amino acid to the growing polypeptide chain. You can see now the polypeptide chain is connected to the amino acid brought in by the last tRNA molecule. So once the tRNA molecule passed down its polypeptide chain to the newly arrived tRNA molecule, the previous tRNA molecule exit from the ribosome and goes into the cytoplasm. So this process continues until the ribosome reach its stop codon. Okay. So reading of the mRNA take place from 5' to 3' end. So as the ribosome read the mRNA molecule, amino acids are added to the polypeptide chain until it reaches the end or stop codon. So the last stage is the termination. So termination occurs, as I mentioned, when the ribosome reach the stop codon at the A site of the ribosome. So once the A site reached, or the ribosome reached the stop codon, it accepts a protein called a release factor. instead of a tRNA molecule. So this release factor, instead of adding amino acid, it adds a water molecule terminating the polypeptide chain. So this reaction release the polypeptide chain from the ribosome and becomes a independent polypeptide chain. So once the stop codon is reached, there are 3 stop codons: UAG, UAA, or UGA. So once once the stop codon is reached, instead of a amino acid, the release factor adds a water molecule terminating the or freeing the polypeptide chain. So once the ribosome reaches the stop codon, it disassembles back into the small and large subunits. Okay, let's recap what we learned so far. So here we have a cell. So cells are the basic unit of all organisms, and inside these cells we have a nucleus, and inside the nucleus we have our genome. So in humans we have 23 pairs of chromosomes, and these chromosomes are made out of DNA strands, double helix, tightly bound around histone proteins. And within this DNA we have regions called genes. And when the gene is switched on, RNA polymerase get attached to the gene and start to transcribe the mRNA molecule. So free bases are added to the mRNA molecule as the RNA polymerase moves along the DNA strands. So before this strand used as a template for translation, RNA processing must undergo. So modification of the ends and removal of introns is done at the RNA processing stage. Then this mRNA strand is transferred into the cytoplasm. So in the cytoplasm we have a structure called ribosome made out of small and large subunit, and ribosome reads the message in the mRNA molecule, and tRNA molecules brings in the amino acids. So there are 20 types of amino acids, and according to the message in the mRNA molecule, polypeptide chain is made, and at a given time 3 nucleotides are read, and at a given time a single amino acid is added to the polypeptide chain. So once the ribosome reaches the stop codon, the polypeptide chain is released into the cytoplasm, and it further undergoes modification to form the protein. So translation of a mRNA molecule can be done by several ribosomes at the same time. A number of ribosomes that translate the same mRNA molecule at the same time is called a polyribosome. If a cell just use single ribosome to translate the mRNA molecule, it will take very long time. Therefore, by having polyribosome, the cell can make large number of polypeptide copies very quickly. So as you can see here, this particular mRNA molecule is being transcribed translate by several ribosomes at the same time. So once the polypeptide chain is translated, post-translational processing takes place. Having just a polypeptide chain is not enough for the cell to make a functional protein. Usually, once the polypeptide chain is being made, it undergoes modification to form functional proteins. These produced proteins then can target specific sites in the cell and perform various functions. So, during and after synthesis, a polypeptide chain can spontaneously coils and formed into a three-dimensional structure. These proteins may further require post-translational modifications in order to become active. And some polypeptides are activated by enzymes that cleaves them as well. Other polypeptides come together to form the subunit of a protein. So, if you take a given protein, a protein can be made out of several subunits made out of different polypeptides. There are two types of ribosomes in a cell. We call them free ribosomes. Those are unbound ribosomes present in the cytoplasm, and these ribosomes synthesize proteins that are functional in the cytoplasm, and the other types are called attached or bound ribosomes. So, these ribosomes are bound to the ER, endoplasmic reticulum, and these ER bound ribosomes make proteins of the endomembrane system and proteins that are secreted from the cell. These ER bound ribosomes and free ribosomes are identical, and they can switch between ER bound and being free ribosomes. Polypeptide synthesis always begins in the cytosol, and synthesis finish in the cytosol unless the polypeptide signal the ribosome to attach to the ER. Polypeptide destined for ER or for secretion are marked by a signal peptide. So, polypeptide synthesis always begins in the cytosol. Then, if they are meant for secretion, then these ribosomes get attached to the ER membrane and secrete the or release the polypeptide chain into the ER lumen for further processing and later transfer to other locations. Okay, let's go back to that pea seed example. So, pea plants can produce both smooth round seeds and wrinkle seeds, and these wrinkle seeds taste sweeter than the round seeds. So, there's a enzyme called SBE1, starch branching enzyme. This SBE1 enzyme, when it's functioning properly, it can build starch. The job of SBE1 is to add branches to sugar chains, forming branched starch molecules. So these branches help starch to grow very large and break down quickly when plant or the seed germinates. However, there's a mutation in SBE1 in some chromosomes. You know that in each diploid you have 2 chromosomes. In some chromosomes, what we call the recessive chromosome, there's a mutation. So this mutation prevents the formation of this SBE1 protein. Therefore, recessive plants, when they are homozygous recessive, they cannot produce branched starch molecules. Therefore, instead of starch inside these seeds, sugar starts to accumulate. Since sugar can attract water, during development, these pea seeds swells, and once they lose their vascular connection during drying, they wrinkles as they mature and dry. So you end up with wrinkled seeds. So in the recessive or the homozygous recessive plants, we get wrinkled pea seeds because the recessive gene or genes or the mutated gene cannot produce this SBE1 gene, which branch starch molecules during seed development. However, in homozygous dominant, you get normal seeds, and in heterozygous plants, still you get normal seeds because there's at least one copy of the gene which is functioning normally, which can produce SBE1 protein. Therefore, heterozygous plants can also produce round seeds. So now you can see how genes contribute to phenotypic variations in organisms. This is just one example.